Hello and welcome. Today we are going to start a brand new topic, module number 11, and this is quite interesting because here we will be discussing about the application and emerging research area in neurophotonics. As you know, neurophotonics in itself is quite cutting edge, quite new. So, I decided that here let me just give you a glimpse of some of the topics that are futuristic, in nature, in the sense that these things are not yet evolved, but has the potential, has the capacity to change, modify and even challenge our existing understanding on how the brain works. So, several of the topics, this is by no means an exhausted list, this is by no means to say that these are the only emerging research areas in neurophotonics. I have chosen of a plethora of different types of cutting edge present day research topics on neurophotonics. I have chosen just 4 or 5 of them, and I will be discussing 5 of many in the session that is going to be module number 11. Remember, several of these topics are yet unexplored or minimally explored. So, I will be giving you thus far what we have achieved. Okay. This is by no means a completed research topic where we started with this was our objective and this is what we have achieved. No, it will not be like that. It is simply stated that we are trying to understand how the brain works, how we can modify, how we can modulate brain, how we can see, and this is thus far we have reached. There are, as they say poetically, miles to go before we sleep. So, that gap, I am hoping that remaining path you and I will travel together, right. So, in today's topic, let us talk about optogenetic modulation of neural circuits. So, we start with something that we already know, that we have already discussed, and you know what this is. We have discussed in module number 10 that optogenetics is a cutting edge biological techniques that involve using light to control the activities of cells, particularly neurons in living organism. I discussed that there is going to be a difference between optogenetics and neurophotonics, though optogenetics deals with light to change, modify genes. Thus far, most of the time light has been utilized, or optogenetics term has been utilized for controlling the genes present in neurons, nerve cells. There is nothing preventing of using optogenetics to control the activity of any other cell, right. But in 2005, the mammalian neurons were first fired, first modified using these kinds of white light source inside the mammal's head, mammal being the mice here, the specific red eye mice, and we wanted to try and see how we can modify genetically, how we can modify the different genes, and thereby can we modify the behavior, of the organism. So, obviously again just to repeat from previous lecture, what are the advantages of light based approach? We can offer multiplexed experimentation, meaning you can have a plethora of different wavelengths of light either being passed through a same optical fiber, multi-mode fiber, which have different wavelengths, and you can trigger different section of the brain. Different section of the brain are attached with light activated protein, different types of light activated protein. Protein A is activated by red light, protein B is activated by blue light, protein C is activated by green light, and all of those light activated proteins are somehow at different places of the brain, some in cerebellum, some in cerebrum, some in medulla oblongata, some at frontal and dorsal lobe. By this time you should know what I am talking about. We have discussed this in previous lectures. If you are having difficulty, just go back to the previous lecture and you will see what I am discussing. So, you can, it's up to you whether you want to modulate or fire one single neuron or one single group of neurons or present in one specific part of the brain, or you can have multiple neurons firing simultaneously but separately, and then look into the combined effect that has on the behavior of the organism, right. Problem with electrical signals, when you put electrode inside the head of mice and then pass electric current, the electric current affects, if not the entire brain, almost a large portion of the brain. So, it will be very difficult for you to differentiate between which part is conducting what exact sense. In case of light, when you have 4 or 5 wavelengths simultaneously, you can give each one of them individually, combination of 2, combination of 3, all 4, and in all combination. Electronics engineers, 0 1 0 1 0 0 0 0, like the truth table, A, B, C, D is the input, A means green, B means blue, another is yellow, another is orange, and then 0 means fire, 1 means not fire. You have the full truth table 2 to the power n, 2 to the power 4, and you have the full truth table and the output, just like logic gate. Just like logic gate, you can do this and thereby try to figure out what is the behavior, what is the combined behavior, what is the overall behavior of the organism. We obviously combine fluorescence imaging with optogenetic manipulation. Multiple optogenetic actuators with distinct spectral sensitivity can be used. The sensitivity is a big part. We can increase or decrease the sensitivity by adding different type of fluorophore. Light is activating the protein, but how much? How much of what intensity and how much of the light is going to activate? The same neuron will react differently, will fire, the electrical channels will be activated, the resting potential and the action potential will be different based on how much intensity of the same light, of the same wavelength have been utilized to fire it, right. We can control it, same wavelength of light, same protein, but the amount, amount is increased, 220 volt, 400 volt, 600 volt, they all have different effects. So, combine the same thing, utilize the same thing, that multiple optogenetic actuators with distinct spectral sensitivity can be used. Proper calibration and control are necessary for successful multi-wavelength experiments. So, you have light electrical simulation, everything is on, light simulation, and you have optogenetic simulation where normal neuron with CH2 channel rhodopsin, some of them are on, some of them are off. Green means on, gray means off. So, you can selectively describe which portion of the neurons are on, as in case this will only happen when we have light activated protein channel rhodopsin, as compared to when none of the proteins are light activated or all of the proteins are electrically stimulated, everything is on, right. So, what exactly are optogenetic actuators and their engineering? So, optogenetic actuators are basically proteins engineered from natural form to respond to light in different system. I told you there are several jellyfishes, which show bioluminescence. You must have seen it in Instagram reels and places that people go to some specific beaches, and at the night because of the presence of jellyfish or some kind of planktons, basically called phytoplanktons, they glow. You must have seen it in different Instagram reels and social media posts, etc. So, there are these jellyfishes, there are these sea creatures that themselves contain proteins, they themselves contain genes to manufacture these proteins, express these proteins inside their body, which glow when excited by specific form of light, right. and this glow, although faint, is usually not seen during sunlight, but when the low light conditions, especially during evenings and at the night, they simply glow up. If you have not seen this happening at different beaches, just do a quick internet search, glowing beach in some place in I think Southeast Asia or I think Latin America, Caribbean, and you will see. So, what the scientists did, that they were able to extract that particular gene. The light activated protein manufacturing gene were simply extracted, and that was placed inside the brain or inside a mammal, right. So, optogenetic actuators are engineered proteins from natural forms to respond to light in different systems. They do not exist naturally in mammals, most normal mammals. If there are a substantial variety of mammals, something might be coming up in days, but usually mice do not glow in dark. Yeah, usually mice do not glow in dark. So, these actuators that are engineered proteins are adapted for expression in various environments to enable light based cellular manipulation. Most engineered proteins are not intrinsically light sensitive and require a cofactor for functionality. Cofactor properties determine the energy and wavelength of light needed. Some protein like GFP, green fluorescence protein like DROPNA and UV light receptor do not need cofactor for activation. So, what exactly is a cofactor? Cofactor is a helping molecule. Cofactor could be an organic or inorganic molecule which is usually not protein. Cofactor, consider it some kind of like a fluorophore that is connected with a large protein and which itself is getting activated by the light and which then creates a biochemical reaction into the overall protein and the process starts, right. So, there are certain proteins which are directly activated by light, and then there are certain proteins that need some sort of a catalyst, some sort of a extra additional molecule associated with it. This extra additional molecule, helping molecule, are called cofactors. So, actuators are the engineered proteins and cofactors are those helping molecules that are attached with the actuators to make them react in a specific way when light falls onto them. The actual photochemical reaction happens because of the cofactor. This cofactor triggers a particular effect onto the actuator. Now, you might ask why this is necessary, or why don't we use all proteins which are light activated by themselves. Yeah, there are certain proteins, just as I was saying, but you do understand that it is simply not that easy to extract a gene from jellyfish and simply insert into a mammal and think that the behavior of the mammal will be exactly the same. Yeah, you do have to bring into some sort of a compromise. You cannot simply start inserting XYZ gene from any XYZ living organism into any other organism. Yeah, it will simply not biologically viable. So, there are certain limitations that you have to follow. Certain proteins like GFP etc., you can directly insert using human body, mammal body. We still have to get the ethical permission to do these kinds of things in human body, especially in their brain. But then we can put some normal protein, existing protein, non-harmful protein, tag it with another molecule which is itself photosensitive, right. So, two ways: you extract You engineer proteins that are photosensitive. Either you can engineer proteins that are directly photosensitive, but have limited value, how much photosensitive, how much energy, how much electrical field, how much ions they are going to trigger, what kind of biochemical reaction that is going to happen, or you can simply tag, you can simply label certain existing proteins with a cofactor. With a cofactor, the cofactor is the one which is getting attracted, which is getting stimulated by the presence of light, and it creates some sort of a biochemical reaction which starts sending signals, biochemical reactions, etc., into the overall protein, which in turns affect the overall neuron cell, which is basically made up of that. So, what are the different types of cofactors that we can use? Remember, these are non-protein. These are non-protein molecules. They can be polyenes, aromatics, or tetrapyroles. Sometimes metallic nanoparticles have also been used. We will be discussing metallic nanoparticle pretty soon in brain. So, polyene binding rhodopsin, we have discussed rhodopsin before. So, rhodopsins which binds polyene in retinal are widely used in optogenetic applications, especially something related to when you are trying to understand the property of vision, visual cortex. Remember, visual cortex were at the back of your head. This includes light gated ion channels, pumps, and G protein coupled opsins. G protein are guanine nucleotidase proteins that are some sort of a enzyme type things that binds the nucleotides. Aromatic binding photoreceptors, aromatic cofactor flavine is used by phototropins and FAD photoreceptors. These proteins respond to blue light. This aromatic cofactor, aromatic molecule, they are specifically for blue light, and tetrapyroles. Pyroles, probably you know, are very common these days. Pyroles are these long ligand molecules, long chain molecules used in enzymes, to solar cells, to supercapacitor. Chemist will probably know. So, tetrapyroles, these are long chain molecules type of things. They are binding proteins like phytochromes. Utilize Blinstead cofactors and responds to light and affect cellular processes. So, if you are not, you do not need to go into too much of chemistry into this, but understand that there are usually 3 different types of cofactors. Cofactors are non-protein molecules that attach themselves into the protein, and they are the one which are directly photosensitive. They are directly affected when light falls into them. When light falls into them, they start some sort of a modification, some sort of a change, which results in the overall change of the molecule of the protein that they are attached with. You know about domino, right? You know, several cards or several matchboxes all lined together. You just drop one and it starts, every other matchbox starts falling. You have seen this numerous times in social media. So, it is like that. You affect one part of the molecule which is attached with a long complex protein chain, which is then connected with the cell, and you just hit one with a pulse laser source, and the biochemical reactions get triggered. It results in action potential, resting potential, all of that firing, and you can clearly map it either behaviorally or by doing all those microscopic techniques. So, cofactors are the one that starts this domino chain reaction. So, G proteins have recently got quite some traction. G protein, I actually forgot, it's I think guanine nucleotide protein, but do correct me if I am missing something in between. G proteins are guanine nucleotide protein. So, G protein coupled optogenetics, they have come up with a very interesting name, OptoXR, GPCR, G protein coupled optogenetics. R stands for, I do not know what it stands for. It often referred to as GPCR based optogenetics is an approach that involves utilizing G proteins coupled receptors, O are for receptors, to modulate cellular activity in response to light. So, as I said, not only it will respond to light and do the biochemical reaction, that biochemical reaction will result in some sort of modification in the overall cellular process, right. There are plethora of cellular processes that keeps on happening. So, GPCR, it is interesting that the cytoplasmic domain of GPCRs are engineered to respond to light, allowing researchers to control intracellular signaling cascade using light stimuli. So, let us try to understand this. So, you have produced or you have created some sort of a protein into the cell wall, right, into the cytoplasmic membrane or into the cell wall, and depending on whether this protein is activated or not activated, you can open or close certain membranes, certain pores, certain areas, certain doors of the overall cell membrane. Cell wall is for plant. So, chimeric constructs are created by combining the light sensing domains of specific light sensitive proteins, rhodopsins, with their interacting signaling domains of GPCR. This fusion enables the GPCR to respond to light and initiate signaling pathway upon illumination. So, a door into the cell membrane which is connected with a specific protein, you can, upon your choice, upon shining light onto it, open or close, and thereby allow certain molecules to pass through or not pass through, thereby affect the overall environment, overall cellular environment, change the pH, increase the temperature, change the polarizability. Remember, the change of polarizability between outside and inside results in flow of electric charge. So, how much amount of electric charge will be flowed can be controlled, can be simply modified by opening and closing the channel gate, opening and closing the cell membrane gates, right. You know about this action potential. Outside is Na, inside is potassium ions, and then you are opening the channel, you are closing the channel, and thereby the polarizability, it is hyperpolarized or unpolarized. This is exactly how vision keeps on happening, hyperpolarization, etc. So, you now have in your hand the tool to simply manipulate it according to your own will. Depending on the type of laser source that you are using, depending on the pulse duration, depending on the intensity, depending on the wattage, you can open or close these pores in the neuronal cells, in the neurons, and thereby allow the overall polarizability of the neuron, resulting in either triggering action potential or creating a resting potential. This is extremely cutting edge. Think about how far we have come. So, this is exactly what I was talking about. So, this is channel rhodopsin, is a light sensitive proteins that plays a pivotal role in optogenetics. This applied in 2005, and this comes from this. So, CHR2 functions as a light gated cation channel, allowing the passage of positively charged ions such as sodium and calcium across the cell membrane when activated by blue light. So, this is the gate, you have activated it by blue light, and it allows the flow of positive ions. So, if this was negative before, by adding positive you are neutralizing it, or if was positive before, by adding far more positive ions you are hyperpolarizing it. You are making it much more polar, much more positive. This ion flow leads to change in the membrane potential of the neuron, which can trigger action potentials, electrical impulses of neuronal activity. CHR2 channel rhodopsin is optimally activated by blue light with a peak absorption wavelength of around 460 nanometer. So, by switching on and off, you can make this go inside or simply close it off, and thereby trigger action potentials or not trigger them, right. We also have gene targeting approaches. This I think you have we have discussed. We produce transgenic mice. These are the transgenic terms, means a mice usually have a particular set of genome sequence, you transfer certain genes from other species onto it, making it transgenic, which is genetically modified. You have heard of GMO foods, genetically modified organism food, brinjal. There was a controversy about GMO brinjal coming up. So, transgenic mice is genetically modified mice are genetically engineered with DNA sequences that includes the opsin gene coupled with regulatory sequence. These promoter sequence, promoter is the gene that starts, the start codon sequence drive the expression of the opsin gene. So, if you have an already existing gene inside the mouse which is getting affected by light upon light delivery, then obviously you have better control and there is no point of adding cofactors, etc. You still might need cofactors, but depending on to increase or decrease the intensity. Optogenetic control can be achieved once specific expression is confirmed, brain wide expression occurs, but off target effect should be considered due to connected neural pathways. You use basically a virus to carry the particular gene, which then attaches itself to the nucleus of a neuron. Into the nucleus, the virus inserts its own viral DNA, viral protein, viral nucleic acids, DNA or RNA. That nucleic acid is the nucleic acid which produces opsin. You have engineered this virus as well. You have engineered this virus. Don't you think that virus could be engineered? Think about the pandemic that we just suffered. Anyways, viral vectors are engineered virus used to deliver genetic materials into cell. Several different types of virus, adenovirus, lentivirus are common viral types. Minimal promoter fragments are required. In some cases, viral vectors are combined with transgenic animal for enhanced specificity. What type of laser source do you need? Well, light source do you need? Obviously, there is a laser source that you need to trigger actually this firing of action potential through neurons. Lasers are the best one. Laser provide highly focused, intense monochromatic light. Common laser waveguides are blue, basically to affect channel rhodopsin, etc., blue, green, etc. Lasers are excellent. The main advantage of laser is it's highly focused, intense, and the polarization is properly done as compared to light emitting diodes and arc lamps, but they are far more cheaper than the laser system. Obviously, fiber optics and 2-photon laser scanning microscopy has also been used. We have discussed 2-photon laser scanning microscopy before, and fiber optics, as you know, we can insert the fibers inside. And what are the applications? We map neural circuits. This is the main and significant applications. The entire brain, neuron by neuron, could be mapped in a mammal, and each neuron could be identified with its specific functions, specific group of functions, instead of thinking that this part of the lobe of the brain is associated with sensors and this part is associated with language and this part is associated with attention and concentration. We now can potentially map neuron by neuron what function is working. Neurological disorder treatment is of course there. Behavioral studies, triggering a particular set of neurons allows a particular behavior to be happening. There is a very famous YouTube video in which there is optical fiber connected with the head of a mice, and as soon as the light is triggered, laser is on, the mice is behaving in a predatorial manner. It is becoming violent and attacking anything that is nearby. So, they have put plastic caps or pieces of wood which are not food for the mice, but as soon as the light is triggered, the mice is attacking it. As soon as the light is switched off, the mice is calm. So, you can try to see the behavioral response. See that in if you get a chance, search for it in YouTube video and you will see optogenetics mice, just type that and you will get it. I have not added it here because of course copyright issues, but I can ask people to visit a particular YouTube video. Vision restoration, neural prosthetics and brain computer interfaces, all are the different applications. So, in conclusion, there are several ways in which optogenetics can help us and shown immense potential to map the brain. So, these are the concepts that I have covered today and this is the reference, and I will see you in next class. Thank you.